So welcome everybody to our third webinar on COVID-19. Anybody think COVID-19 seems to be a hot topic in the globe? Today we're talking really around soap and disinfection and cleaning and that type of thing and manufacture. So we'll get straight into it. I'm just going to do a quick, my name's Scott Colborne. I'm just going to do a quick introduction for everybody. Just to set the scene for what we're going to talk about today, and then I'll hand it over to our speakers to carry on through. So, a bit like what we've done before, and if you've been in our ones in the last two, I do apologize for kind of repeating myself, but we've got three speakers, each are going to present for about 15 to 20 minutes and do a quick bit of Q&A afterwards. Then once all of them are finished, we'll continue with the Q&A with everybody on board there. Don't worry if you miss some things, because the webinar is going to be recorded and will be uploaded to the RACI website and to the YouTube channel. We are going to use, because there's a lot of people on the line, we're going to use the Q&A function, not the raise hand function. So on the bar at the bottom or at the top, depending on how it shows on your screen, you should be able to click on Q&A, and please do type in any question you have. The more questions, the merrier. We do have some questions that were asked before the webinar, but it'd be great to have ones that people have already put together. But please do that as we go through, or in the chat window. It doesn't really matter as long as you ask them through there. And all the panelists will be able to answer those for you. So today, the speakers, we're going to have Dr. Greg Whiteley first. Greg is the Executive Chairman of Whiteley Corporation. It's Australia's largest manufacturer of hospital grade and higher level disinfectants and sterilants. He's a well-published researcher and patenter, involved with the School of Medicine at Western Sydney University, and also, commercial research director in investigations into biofilm mediated infections. So pretty interesting stuff. The second talk from Pall is going to be on soap. I think everybody's interested in what soap does and what soap doesn't do with COVID-19. Pally's at UNSW, an expert on supramolecular, if I pronounce that correctly, supramolecular chemistry and self-assembly. Viruses are actually self-assembled nanoparticles, and he has been consulted by the media already around how they might destroy detergents, detergents, and that might destroy the virus. Then lastly, we've got Damien from Dominant, also with Amber as well as the marketing director, talking around the challenges that they've seen there during manufacturing at their company, some of those regulatory and compliance things that we see there. So that's the three talks we're going to have, and I'll kind of stop talking now and I'll stop sharing my screen. I'll hand over to Greg to get us going.
Thanks very much. And I'll hit this and hit that. And away we go. Yes, my name is Dr. Greg Whiteley, and thank you, Scott, for the introduction. I'm going to be talking more generally around disinfectants, large and small, and particularly focused on efficacy and safety considerations. One of the important things to note in this area is that there has been so much rubbish in the media. It'll be nice to actually talk about evidence. Now, before you even consider using a disinfectant, of course, one question to ask yourself in every occasion is, are the bugs dangerous, normal, or even useful? That applies to human disinfectants, that is skin disinfectants and antisepsis, in terms of infections in body cavities, which is something we're particularly interested in, and the presence of bacterial biofilms makes this whole area very interesting, and the field is now quite dynamic. The interface between what the microbes are doing and what the disinfectants or the antibacterial or antimicrobial solutions are doing is particularly dynamic, particularly in live people. An inanimate surface tends not to fight back, even if it's got an antibacterial or antiviral coating, but humans and animals actually do have immune systems. So the whole environment is dynamic, and that means that we've got a constantly changing plethora of chemistries going on. And the most important thing too, as a third part of the introduction, is to say that the simple things are the most important things with COVID-19. things like making sure that you've got clean hands, that you're wearing gloves if you're going to do a cleaning process or a disinfecting process. Sprays are fine, but you can use sprays or squirts, and then wipes. Unfortunately, in today's talk, we don't really have a chance to deal with the whole issue around cleaning, which is another subject in itself, but we'll pose some questions at the end. Now, as we're all aware, COVID-19, as this comic sets up, has really hit the streets. And despite the comedy of this, there's been a substantial global death toll. And we've been remarkably spared from a heavy death toll in Australia. I note even today in the Australian Financial Review, former Senator David Leyonhjelm expressed a contrary view to taking control in the way that the federal government has done. But we are where we are. And I think you've already had some fairly good lessons about what this virus does and some of the funny little things this virus does to make it such a potent pathogen and why we are in a pandemic. The two of note are, first of all, asymptomatic or pre-symptomatic transmission. That is to say, there is large viral shedding going on prior to the onset of serious symptoms, which means you can be infected by someone who has no outward signs of this viral infection. The second thing is, there is no doubt this virus does survive on surfaces, but one of the questions in this area, and I think Pall is going to deal with this as well, is when you do this disinfectant work in the lab, you're working with pure virus, whereas when you get in the real world, the virus is almost never pure virus. It's always present with other materials. And we'll look a little bit at that as we go forward. Another area, by way of introduction, that's an important point to make is that disinfectants are classed in Australia as medical devices. So that's one part of the therapeutic regime. Whereas antiseptics, antiseptic hand rubs, antibacterial hand washes, skin cleansers, anything that's used directly on the human body is regarded as a medicine. And so all of the products that are medicines are strictly prohibited from making general virucidal claims unless there's been a full therapeutic trial, clinical trial conducted with clear outcomes. So things like your standard antibacterial hand rubs will never have antiviral claims on them, unless of course they're illegal, but we'll come to that in a minute. And that's important because people wonder about the efficacy. Well, the problem for manufacturers is even if we have the data, we're not allowed to talk about it, we're not allowed to put on labels, we're not allowed to put it into any form of literature. We can hold the data, but we can't actually promote that. And of course, that's important, and I know Pall is going to deal with soaps and so forth, but really there are two basic approaches to hand hygiene. And the first one obviously is washing your hands under running water with a soap solution, whether that's a natural soap or a synthetic soap, I won't get into here. Or the alternative is one of these antibacterial hand rubs, some of these medicines. There's another group of them that are not regulated, where they've got claims limited. They also don't have to be made under therapeutic manufacturing conditions. Those products are widely available. They can be identical in terms of their formulation, but the nature of where they can be sold is quite different. Only products that are registered with the Therapeutic Goods Administration as antibacterial hand rubs or antibacterial solutions can be sold into healthcare settings and to healthcare professionals. One of the funny areas, and I know Pall is going to deal with this, is there's been some extraordinary speculation about what works and what doesn't work, and some very uninformed, totally non-evidence-based media commentary in this area, which has actually been very unhelpful. And the TGA is now starting to clamp down on that, and we'll go through why and how they're doing that. Now, some of the evidence that sort of lacks, or methods that lack evidence, this is a great example. Here's somebody coming along with some magic spray gun in an aeroplane, hoping to disinfect all these surfaces with an unknown solution. Interestingly, the PPE is insufficient in this case because there's clearly a spray, there's exposed skin, there's not proper goggles. But of course, it all gets worse. And we now have not just crazy methodologies going on, but also snake oil everywhere. You also have the ridiculous overreaction to things. The reality of this virus on the surface is that you certainly can recover the virus for long periods after people have moved out of the area. On one of the cruise boats, it was viral recovery 18 days after the room had been vacated by a patient who was positive to coronavirus. So that's a fairly long time. But the infectivity is a different issue. And there's some very good papers that have been published on virus survival on surfaces, and some of that have been complete rubbish. I know Dr. Trevor Glasby and myself have written a number of letters to the editor of international journals, making the point that in a couple of cases, some of those articles that have been out there about virus survival have actually had no effective peer review. There was one in a very reputable journal, the Journal of Hospital Infection, which is widely cited, and yet peer review was not at all done. It was reviewed by the editor and accepted for publication on the same day as it was published. And then when you get into the actual evidence presented, there was no evidence. It was a compilation article. The underlying literature on which it was relying was misquoted or severely curtailed, so that the actual message of the underlying papers was not presented in the final paper published recently in this other journal. So getting into the evidence is a bit of a problem. And one of the problems here is that disinfectants in Australia are regulated. These are quite well controlled by the Therapeutic Goods Administration. That series of controls have been in place since 1996. It's a Commonwealth statutory authority, which means it has governance over the entire Australian territories, states and territories. The core legislation under the original Therapeutic Goods Act is Therapeutic Goods Order 104. That's fairly recent. It replaced a prior piece of legislation that had been there since 1996. There is a second piece of legislation which picks up sterilants and instrument disinfectants. We'll come back to those. That's under the Medical Devices Regulations of 2002. So the legal complexity here is not to be understated. And the important thing as we get into it is that there is quite substantial framework around how this whole process works. TGO said at 104, as did TGO 54, sets out classifications for disinfectants with associated signal headings. That is to say, the signal headings are indicating what level of performance is required. So an instrument disinfectant is clearly intended to be a disinfectant of instruments. A sterilant intended to sterilise, whereas a hospital-grade surface disinfectant is intended for surfaces. And the risk of each of those is graduated in the performance standards set out under the legislation. One subtly about this is that because the microbiocidal performance testing is standardised, it's very hard to see much of the information that's out there in peer-reviewed literature, because frankly, it doesn't meet peer review standards. If you're just throwing a new disinfectant with a moderately different formula and you're throwing it up against standardized disinfectant tests, it really is not going to meet the requisite standards for innovation. So there's a large body of information held actually in industry. Much of it is protected by individual companies because there's obviously commercial and confidence issues. That will include products that fail or formulas that fail as well as those that do well. But it's an important point to make, because if you're going to use a disinfectant, it does throw the emphasis back on interrogating the company or the supplier for what that evidence is, because you generally will not find it in the literature. Here are the basic framework signal headings for disinfectants for human applications in Australia. Obviously, sterilants at the top. You've got instrument disinfectants. There are 3 separate standards depending on what you're trying to disinfect. For example, a gastroendoscope, which goes into a mucosal surface, will need to be disinfected with a high-level disinfectant, whereas something that was being used on, say, intact skin, you might take a stethoscope, you only need low-level instrument disinfectant. For sterilants and instrument disinfectants and instrument-grade wipes, the TGA requires full manufacturing licensing of premises. So there's a whole TGA approach to licensing of manufactured premises, whether they're in Australia or elsewhere. Hospital grade disinfectants formerly were all on the ARTG, but the only hospital grade disinfectants that are still kept on the Australian Register of Therapeutic Goods are those with additional claims, such as for viruses. And then you've got wipes, commercial or household grade disinfectants, and then sanitizers. Now, important point to make is that a hospital grade disinfectant that just wants to claim bactericidal performance has a limited range of claims. There's no need to register those products with the Australian Therapeutic Goods Administration. However, if you wish to make a virucidal claim, whether that's for Covid19, SARS-CoV-2, or whether it's for influenza virus, or whether it's for herpes simplex virus, every virus that you're claiming to disinfect will require that product then to go back around through the TGA system, and all of the evidence so far as the claims will be examined as part of a registration process prior to you being able to use those claims on the label. Another subtlety of this whole process is that to advertise any of these products in the public domain, that is not just to healthcare providers, requires explicit Therapeutic Goods Administration approval for the advertising material, including the exact wording that's going to be used. So there are quite a deal of complex layers of controls in this marketplace. Now, when it comes to coronavirus testing, generally we're using surrogate viruses. I was personally involved in the negotiations to make sure that we could use viruses in Australia. The one that most people are using in Australia is the murine hepatitis virus, and human coronavirus 229E is becoming available, but I'm not sure that it's quite here yet. But certainly the TGA quickly recognised that those two viruses were suitable surrogates. We do use surrogate viruses on a number of other areas. So the hepatitis group viruses, we use surrogate viruses. You can also use a surrogate virus for HIV testing. There are a few other places where surrogate viruses would be an appropriate way of getting claims that were suitable. Now, in Australia, the TGA basically has mandated that only products that have been tested against one of those two viruses will be allowed to put a COVID-19 claim on their label. Now, that's very different from, say, the US EPA, where they accepted broadly that if you had evidence that your product was virucidal against another envelope virus or one of the more difficult to kill viruses, you were automatically recognised as being acceptable to put a COVID-19 claim on your label. Consequently, there are over 370 individual product entrants on the US list end. There's a website pointer you can go and have a look at. Here's the list of raw materials. You'll see that it's a bit of a who's who of the disinfectant world, including a whole array of acids, all the different versions of chlorine, some oxidizing materials, quaternary ammonium biocides with their single chain quats, dual chain quats, or any other combination, the full array of alcohols. There's some phenols and some other essential oils that are in there. But if you look at the vast bulk of products that are on that list, 49% of the products use quaternary ammonium biocides as their primary disinfecting molecule. Now in Australia, the list is concomitantly much smaller. And if you look at the statistics and take the ratio between the two on the basis of population, it's about right. There's 27 approved products in Australia. There's the pointer for anyone that wants to see what products are currently on the ARTG. And look, that list is expanding as more and more people are getting their products tested, and those products are then entered onto the Australian Register. One important point about the, just stay there for a moment, about all of this testing is all of the disinfectant data where you're submitting it to the TGA must be done before you start selling the product and making the claim. And it must be done fully independently, and it must be done on an approved or standardized test method. And there's quite an array of those, depending on The risk class of your product will depend on how many tests that you will need to do. So on a sterilant or a high-level instrument disinfectant, you could be doing anywhere between about 22 and 27 standardized tests to prove efficacy. But it's important to note all that testing is done at independent laboratories, and in Australia those laboratories are also themselves normally subject to TGA audit. So there's both a feedback loop and a control loop. For overseas laboratories, there's a little bit more latitude, but most people are ending up with laboratories that are either supervised by the US FDA or basically somewhere in Europe through one of the other authorities in Europe. Now, of course, bug zapping is all the rage at the moment, and the snake oil people are out there with claims. The TGA are out there actively in the marketplace at the moment, basically beating up anyone who's making false claims. And there's good reason for that, because when you look at how disinfectants work, there are trade-offs. You've probably all seen this before, but there is a clear three-way trade-off. You only ever get 2 points on the triangle. My favorite example is hot water. Water's a terrific disinfectant when it's boiling, but of course it's quite dangerous to your health. If you pour it on your skin, you'll get burned. On the other hand, if you reduce it to tap water and at potable water temperatures, normal room temperature, then of course it won't disinfect. Another critical series of factors is these three things must be put into place for a disinfectant to work. Direct contact. If you don't have direct contact, these materials do not work by remote control. You can't get out the Harry Potter's oak wand and wave it around. You must have direct contact between the chemistry and the microorganism. And that applies equally for coated surfaces where they're making claims that you never need to use a disinfectant. That's just complete rubbish, because surfaces always get contaminated. And the moment you have any sort of physical barrier, you will therefore lose the potential for disinfection. The next thing is, of course, time. These products will take varying lengths of time. Something like alcohol is going to be probably in the range of 30 seconds as it dehydrates the cells. Phenols, on the other hand, which usually lyse cells, are almost instantaneous for those bacteria they're going to kill. And temperature is the other thing. We work on a principle that about a 5-degree change in temperature away from room temperature, whether it's up or down, will cause a factor of 2 in terms of the exposure time required. And of course, the bugs hide. We have to remember, these bugs are not wanting to be killed. Evolution tells us they want to survive. I mean, the whole reason this Covid19 bug has hopped out of an animal population is it's making sure it can survive. It's found a new host. It's doing something different. That evolutionary biology has great relevance. Now, when we come to doing the disinfectant testing, we therefore need to be aware that you're doing 3 basic categories of testing. One is an in vitro test without anything that's there, just purely the bugs. Then a disinfectant in vitro test with soils. And then there's also simulated in use tests. And the sort of soils that are used, normally, obviously hard water, organic soils, serums, oils, carbohydrates. It depends on the test you're running. Cellular materials such as blood or blood and cellular fragments. It's relevant for the blood-borne viruses, even the surrogate viruses. And then, of course, we've got more recent work on biofilm methods, and we've certainly pioneered a whole pile of that through various ARC grants. And some of the photographs I'll use in a moment are from Associate Professor Karen Vickery at Macquarie University, who's worked with us for nearly 30 years now. Now, what do you clean and disinfect? Well, the primary thing to aim at is not this whole spray, it's what people touch. Because one of the features of this virus is you can self-inoculate. That is to say, if it gets on your fingers and you wipe your nose or your eyes, work done by Professor Mary Lou McLaws at the University of New South Wales showed that most people touch their face between 20 and 40 times every hour. And so if you've got hands that are contaminated because they've touched something that's contaminated, the chance of you moving the virus from that surface onto your fingers and into your eyes, nose or mouth is fairly substantial. Work we've published in the last 3 years has shown that when you get one of these bugs, well, we've mainly looked at bacteriology and we're looking primarily at Staph aureus because it's got great survival characteristics. It's a standard nosocomial infection. and it's got great robustness. But Staph aureus can stay on your fingers or your hands or your gloved hands for up to 19 subsequent touches. So the bugs stay there, they don't just go away. And the problem of biofilms, which we've been investigating now for more than a decade, is really not to also be underestimated. We don't know what the role is with this virus if it comes into the hospital environment with biofilms. We suspect from what's happened in places like Northern Italy and also parts of the US where they've got biofilm problems in hospitals with things like Candida auris, let alone the bacteria, that there's probably some surface involvement, but we just can't quite get to that yet. We have an active grant at the moment in place, waiting to hear how we go, looking at exactly that. Here's some micrographs from Karen Vickery, looking at different biofilms. On the top left, that's from a paper published in 2012 in the journal Hospital Infection. Some of these others are various papers. On the top left, you'll see there's 4 little micrographs. The one on the bottom right-hand corner, you can clearly see a piece of rayon on a blind cord. The one underneath it is from an ultrasound pillow, and you'll see there's various bacteria as well as the biofilm mass. On the top right-hand corner, you've got a piece of fibre off another cord. And again, there's a small nick in the fibre and there's biofilm in there with various rods and cocci. And on the bottom right-hand corner, from Scotland, again, is fabric from a chair with coccoid and other coccoid and other bacteria and biofilm present. And these biofilms are where you touch, and that's where the problems are. Now, unfortunately, we don't have time today to really get into issues like wiping, which you would think is something simple, but in reality, there's at least 9 variables involved in wiping. So the whole area of cleaning is something that we're, after a long period of latency in looking at how this works, and we're really pleased at Parley's work on soaps, because it's going to revisit a whole pile of fundamental cleaning work that's not been looked at for the last 50 years, and we need to go back and look at it. Work that I've done in my own postdoctoral area has been looking at how we measure cleanliness on surfaces, how you do the validation. I've focused on things like rapid ATP measurement, and there's some real benefits there. There's some real downsides as well. Again, unfortunately, we can't deal with that today. And I'm going to hand back to our facilitator, Scott, and thank you all for listening.
Thank you, Greg. That was wonderful stuff. Thank you. Our participants are a little bit shy on questions at the moment. We've kind of got one through, so I might just do a quick one with you. Right at the start, you talked about surfaces in the lab environment. So really asking, is it not as potent on surfaces compared to when you kind of study it in the lab environment? Is that something?
It's actually the opposite.
Oh, okay, okay.
When you look at it in the lab environment, you've got pure virus. And I mean, the virus has certain survival characteristics. And if you look at, there's a paper by Van Doren Marlin in the New England Journal of Medicine, which is well cited. It's quite good work where he looked at a range of materials and compared SARS-1 and SARS-2. And you just need to look at how the different viruses survive on different surfaces, and there's significant time differences. My favorite is SARS-1 survived only 4 hours on cardboard, and yet SARS-2 managed to get to nearly 24 hours. Well, that's a big difference. And that's just putting on the environment. If you then surround it inside, say, human spit, when we expire the virus, it's not coming out as pure virus, it's coming out with nasal aspirates or oral aspirates. And so it's in a hydrated solution, in a droplet. These droplets, the smaller they are, the more they'll stay suspended. Most of them fall out fairly quickly. But when they're on the surface, they're in droplets. It's not pure virus. It's very hard to get that fully replicated in the lab. So it's actually the opposite. How will it go in the real world? Bit of a question.
Yeah, I think unfortunately people want a set number, don't they? If I leave it 1 minute and 1 second, I'll be all right. But if it's 1 minute, it's not. And life just isn't that simple, is it? Unfortunately,
life is not meant to be easy. Wasn't that famous?
I think so. My mother told me once, if everything was easy, everybody would try it. But anyway, I've got another one through. So you mentioned quats. did that mean they all work against COVID-19? Does that make sense to you, that question?
Yeah, it does.
Yeah, good.
It's concentration dependent and time dependent. I don't want to steal Paulie's work, but you know, the quats are a really good disinfectant for a whole pile of practical reasons. They're not as strong as something like the oxidizers, but they're easy to formulate. They're easy to use. They're generally going to be better on the health, safety, environment issue. They're quite biodegradable. You can set the pH and conditions for them to really not present the sort of level of hazard. They don't have a lot of odor. In fact, you can add perfumes to them, which we all do. So cleaners like them. One published paper we have, which was done in Geelong through one of the academics at Deakin University, looked at one of our products versus chlorines. And the real issue wasn't the disinfecting performance in the lab. It was what happens in the field. And people tend to use products they like better in a more effective way. So when it came to the epidemiology, the outcome was there was much better outcome with the quats, even though we know the chlorine should be, you know, equally as good. But they didn't like using it. It smells, stains everything, gets on their fingers.
Yeah, those, you know, unexpected biases that come in, pretty bad sometimes, aren't they? What we'll do is we do have a few more, but we'll move on to Pally's talk, and then we'll gather those questions for the end, so we'll still come back to them. So, you ready to go, Pally? Did you want to share your screen with everybody?
Yep, I think I'm ready. Just the next thing I need to do is to share my screen. Make sure that put it on presentation.
Looks good.
You can
Looks
see
good.
that?
Looks good.
Thank you. Yeah, thanks for the invitation. I'm extremely excited to get to talk to you about this and the journey I took. And I was fascinated by Greg's talk here too. Touched on some of the things that I've been learning actually over the last 2 months. That I jumped in this sort of from a almost theoretical point of view, and I guess like everything else in life, this is just so complicated. And one of the things I find very complicated is the terminology and the nomenclature is not even clear. When someone says disinfectant or says a surfactant, aren't we really more or less sometimes talking about the same thing? Or my classical example is soap or the detergent. Detergent or soap, chemically speaking, I'm not entirely sure how we're going to divide the line between the two. They're basically the same thing. So, as a matter of introduction, I come from UNSW, and I just want to highlight the wonderful people who I've had the pleasure to work with every day, and the people who supported me. I come from this corner of chemistry we call supramolecular chemistry. Been around for about 40, 50 years, and somewhat related to organic and inorganic chemistry. But it's all about trying to understand the role of non-covalent interactions in chemistry, intermolecular interactions. And in my own work in particular, I look to nature for inspiration, because there's sort of two forms of supramolecular chemistry that really matter in nature. One is molecular self-assembly, and that's how molecules self-assemble together to form functional structures, such as a DNA double helix, or maybe more interestingly, how a peptide chain folds up into a functional tertiary complex, and then they come together in bigger complexes. The other aspect of supramolecular chemistry is molecular recognition, host-guest chemistry, which, again, biology you see in the form of transportations and enzyme specificity and so on. And for me, supramolecular chemistry is the chemistry of life, because the cell is made out of, well, relatively simple building blocks like fatty acids, DNA, amino acids. It's really how all these molecules interact that brings life to life. Now, on a slightly practical point and not, one of the things I worked a lot on the last 10 years is polymersomes. These are self-assembled nanoparticles, so they self-assemble from block copolymers that essentially are like soap molecules, except much bigger. So you still have, like in a soap molecule, a hydrophilic and a hydrophobic end, except now we have a much larger polymer, and they self-assemble into these sort of structures. Now, these sort of polymer structures are often, in fact, the same sort of size scale as viruses. So I've always been fascinated by viruses anyway, and the sort of stuff we do in nanoparticle. In fact, I haven't done it yet, but people have even managed to stick RNA inside these sort of particles, so we're getting scarily close to making some sort of artificial viroid. And one of the reasons why we like working with them is that polymersomes are more kinetically stable than liposomes. Now, liposomes are used in drug formulations, and they are made out of small amplifiers, so they're more cell-like. And just to show you, I mean, this sort of stuff, this is work with Martina Stencil and others. Our nanoparticle, actually, the way we designed them, they're capable of penetrating into cancer tumors very effectively. Right, so then this happened. 2020, we will never forget that year, I think, for a while. Not for good reasons necessarily. Got the toilet paper back, at least where I live. And the SARS-CoV-2, the novel coronavirus, is a nanoparticle. And on the left here, you see a transmission electron-microscopy image, one of the first ones actually taken of this virus. The diameter is around 100, 150 nanometers. And on the right is a textbook schematic of a coronavirus, but they're all more or less the same in terms of the overall structure. What you got in the center is the sort of the capsoid, the protein wrapping around the RNA, which is genetic material. And then that's coated with a fatty bilayer, the membrane bilayer, with some protein chains, and then the infamous spike protein sticks out of that and is anchored into the lipid bilayer. So this is, as you'll see in a moment, this is the weakest link in the viral structure. And the cell replication of the virus in itself, I call it a masterclass in self-assembly, because what you have is the virus through the spike protein binds to the cell, gets into the cell, releases the RNA cargo. The RNA then, well, the ribosomes take on the RNA. They don't realize it's a foreign one. They start to make the protein that's encoded in the RNA. Some of those proteins then go on to form what's called a replication complex that then starts to replicate the RNA, because otherwise you wouldn't normally get many copies of the RNA inside the cell. And then, of course, the protein, this RNA then goes into ribosomes and makes more of these proteins. And then all this RNA, new proteins, cell assembles, hijacking again some of the apparatus from the cell into new viral capsules. And on the way out of the cell, just to add insult to injury, steals a bit of the cell membrane to make that lipid coat around the virus. And now it's ready to go. And I believe I understood the reason why you have a lipid membrane. It helps with sort of the aerosolation of the virus in the mucus in the lungs. So when you cough or talk, the virus gets ejected. So the lipid is there to help with that process. And actually, I should say, also with internalization here. Anyway, one Sunday afternoon, I decided to, bit for fun in a sense, write down a Twitter thread to try to explain from a chemistry point of view why soap works so well. This was early on in the epidemic, about the 6th of March. I went outside to play with my kids on the trampoline. I came back and the tweet had gone viral, literally. And that sort of kind of changed my life completely accidentally. I didn't realize this was something people wanted to know, try to understand the fundamental chemistry behind why soap might help us with destroying the virus. And it took off. I was approached by media around the world, and it was quite a journey, to say the least. And I learned even what the University of New South Wales is called in French. One of the things that I did was then to work with this organization called TED-Ed to put together a bit of an educational video. And I'm actually going to use that now. This has reached now 1 million views on YouTube. And apologies, Greg, there are some simplifications here on what's going on. I hope you will hear the sound, and someone has to scream out if it's not working.
The coronavirus that causes COVID-19 is one of many viruses whose protective outer surface is made of a lipid bilayer. These lipids are pin-shaped molecules whose heads are attracted to water and tails are repulsed by it. So, in water-rich environments, lipids naturally form a shell like this, with the heads outside and the tails inside. Their shared reaction to water makes the lipids stick loosely together. This is called the hydrophobic effect. This outer structure helps the molecular machinery of the virus break through cellular membranes and hijack our cells. but it has thousands upon thousands of weak points where the right molecules could pry it apart. And this is where soap comes in. A single drop of any brand of soap contains quadrillions of molecules called amphiphiles, which resemble biological lipids. Their tails, which are similarly repulsed by water, compete for space with the lipid.
I'm going to pause there for a second. And as I said, the lipid, just to point out, the lipid membrane of the virus comes from us. It comes from our own cells. So that this is a conventional phospholipid because it's just our own sort of cellular membrane. Now, of course, most soaps that you buy consist of single tail amplifiers like sodium STS, or sodium lauryl sulfate. Or in fact, it's not really that different if you use so-called natural soap. That's usually sodium salt of fatty acids like C12 and C16 fatty acids. One thing I want to point out is that I'm a sort of a physical organic chemist and we like to think in mechanism, and you might argue, well, do we really know it happens this way? Short answer is probably not. We don't really know it, but we have very good evidence. And for instance, down at UNSW, they did a really cool experiment a few years ago. They made a monolayer of deuterated lipids on a particular piece of a surface down at the neutron source. And then they added a solution of amplifiers, which are non-deuterated. And by using neutron scattering, they could see literally how this amplifier was exchanging with the bilayer. So we do have some experimental evidence that when you got a bilayer layer like this and you add soap to it, that's probably the first thing that happens. You get this exchange between the lipids in the membrane and the amplifier. And this is actually also how soap works on viruses. The problem with bacteria, problem with bacteria is that they have, in addition to this layer, they have all the things in their cell walls and cell membranes and double layers and all that. So soap doesn't work terribly well on bacteria.
Lipids that make up the virus's shell, but they're just different enough to break up the regularity of the virus's membrane, making the whole thing come crashing down. Those amphiphiles then form bubbles of their own around particles, including the virus.
And again, this is something we do know how soap works. It removes dirt by basically solubilizing it. But I will freely admit that, you know, whether the soap mainly breaks down the virus by ripping the lipid layer apart, or it starts by unfolding the spike protein and ripping them out, I guess we'll never fully know. But all you need to do is to deactivate the virus. If you destroy the lipid membrane and or unfold the spike protein, even if the rest of the particle is there, it's no longer infected.
This is RNA and proteins. Apply water and you'll wash that whole bubble away.
So that was only part of the video, because in the second half of the video, we talked about how sanitizers based on alcohol work. And it's a little bit different, because what alcohol probably does mainly is to destabilize protein and lipid structures by reducing the effect of the hydrophobic effect. So this self-assembly that holds the lipid membrane together and holds the spike protein in place, essentially they start to unfold and the whole thing falls apart in ethanol. It does turn out that most sort of, commercial sanitizers are essentially just ethanol, and we kind of know that works. But I have to say, after getting into this,
the coronavirus that causes...
what is the real evidence of that soap, in fact, works on viruses? Well, in reality, the best evidence comes, in my opinion, from what I call studies in the field. And so far, we don't have them actually on the coronavirus, or at least on the SARS-2 virus. We mainly have them from influenza viruses and Ebola in a few cases. So we have them from other enveloped viruses. But here is just one example, some German study on an influenza virus. I think it's H1N1, and essentially showing that after hand washing different volunteers after 20 seconds, they could see more than 99.9% reduction in active viruses on the volunteers. I don't think, Greg, and you might want to explain like whether there are some other forms of tests that are typically done, but in the literature I've read, it looks like what I call the in vitro test are normally done what I call in a droplet fashion. You have a surface and you add a lot of active virus onto it, and then you put a droplet of your solution that you're testing on top of it, leave it for X amount of time, and then check how much is left of the active virus. And this is sort of how they worked out that for these enveloped viruses, the coronaviruses, it seems, you need in a sanitizer probably about 60–70% ethanol to disinfect. Now, so far, I've only seen, and I'm not sure, yeah, this is from the Lancet Microbe, a very recent study with the SARS-2 virus, where they did this sort of in vitro test with bleach, with ethanol, chlorohexidine, and a few other things. And after 5 minutes, the virus is completely gone with all of these. Interestingly, with the soap solution, they saw that the virus wasn't completely dead after 5 minutes. But this is where I think there's a bit of a disconnect between some of these in vitro tests and in the real world tests. Because first of all, they said they did this 3 times, and in 2 tests they couldn't see any virus. It was only in the 3rd test they saw it. But I think it's more important to think about that. I've seen also indications that in the influenza virus literature that people got in vitro tests on soap indicating soap doesn't quite kill the virus, but yet in the field studies on volunteers seem to indicate soap works quite well. I think I know why, but I might be wrong. I'm going to propose this to you. Essentially, as a chemist, I look at the in vitro test and it's a closed equilibrium system. You put the droplet on, leave it to interact with the virus for X amount of time, but there's no influx or outflux. And therefore, the virus might tolerate, in terms of self-assembly, that amount of whatever chemical is interacting with it for X amount of time, especially when you're using surfactants. So when you use chlorine, you're doing a chemical reaction. So that's a little bit different. But when you're using surfactant, it's a non-covalent interaction. Whereas if we just replay this part of the video,
those amphiphiles then form bubbles of their own around particles including the virus's RNA and proteins. Apply water and you'll wash that whole bubble away. Those amphiphiles.
Okay, can I stop that video? I think that's actually a really, really important thing when you actually wash your hand, that you are continuously applying soap and you're washing away the stuff continuously. So it's an open system, it's a non-equilibrium system. So all the disassembled stuff is immediately carried away and re-establish a new equilibrium, and therefore the virus can't tolerate that after a certain time. Now, one reason why I think you need to tell people to wash your hand for 20 seconds, and again, people like Marie-Louise McClure has told me that's sort of been worked out by trial and error in hospitals, mainly because you need 20 seconds to be able to cover the entire surface of your hand with soap. We don't really know whether soap needs to be on a surface of a virus for more than a second or two. Maybe Greg knows the answer to that. And the other point about soap, and I know we're not going to talk too much about disinfecting surfaces, and that Professor Mary Lewis says is that when you're trying to disinfect a surface, you probably actually want to start with soap, not because the soap may kill 100% all the virus, but it will remove the dirt under which the virus might hide. And then you bring in one of Greg's disinfectant solutions to make sure you destroy the virus. Because some disinfectants are not very good at dissolving the dirt, so you may need both. And that's sort of the advice that she works with the WHO, they give to the public, that if you want to clean a surface, maybe you soap first and then a disinfectant. And with your hands, you can't use some of the stronger disinfectants because you just can't tolerate them. Many people ask me whether you can pour bleach on your hand, and I don't think that's a very good idea. But again, we don't really have any study to show this, but I actually don't think that makes the type of soap really matters. It's how long you expose the hand or the surface to the soap that matters much, much, much more. So that's all I wanted to say, and just to thank the people who have helped me with my work, and I'll leave it there.
Thanks, Paddy. That's fantastic. Desperately trying to find the unmute button there, so I apologise for my lack of skills with the keyboard. What we might do, just so to make sure everybody can get all the presentation, might jump straight into Damien and Amber's presentation, then do questions at the end, if that's all right.
this stuff.
Looks good, Damien.
And thanks, Greg and Paulie as well. That was great. It was fascinating to see, and it was a very concise summary by you, Greg, regarding the regulatory landscape around hand sanitizers. So my name's Damien Rankine, and this is my colleague Amber Dance, and we're from Dominant. We are going to have a bit of a talk about an honest account of the challenges associated with manufacturing during COVID. So, I mean, what do we do? We are from a company that makes cleaning chemicals, much like Whiteley Corporation, where Greg is from. We've been around since the 50s, so been doing it quite a while. And we're up to about 70 employees now. So it's a family-owned business, local manufacturing here in Adelaide. So the kind of services we provide are formulation and product design. We do everything from that to manufacturing, packaging, and sales and distribution. Anything from liquids, powders, and tablets. And these go into a huge range of industries. So I&I, healthcare, home care, food and beverage production, hospitality, and industrial cleaning as well. So it's a bit about what we do.
Yeah, and this is just an example of some over this COVID period.
I'm sorry, Amber, your speech is dropping in and out, I'm afraid. I think I do apologise that we've been having a little bit of technical trouble here. Do you want to just try again?
Okay. So yeah, these are some of the examples on the bottom of the screen here of our products that have been in high demand during the COVID pandemic. So this includes the hand sanitizers, hard surface sanitizers, and hand cleaners. And they come in a range of different sizes. So we have 500 ml bottles. We have pods that go into dispensers. And we have the larger sizes, 5 litre, 15 litre, and right up to the 200 litre drums of products.
So this will kind of flow into what we're talking about and why we've had so many, such a variety of challenges during this period. So, I mean, a bit about this presentation, just talk about hand sanitizers in general. I'm glad no one's really covered the individual challenges and ingredients. We'll go into a bit of that, a bit about packaging, the issues we've had there. And as you can see from what Amber said on the last slide, there's quite a variety of packaging options that we have to supply. And then a little bit about how this has impacted marketing of the products. So a little bit of chemistry. So there's, obviously this is a RACI webinar with some chemists out there. So the formulation of most hand sanitizers is not, a well-kept secret. It's pretty simple. There's a lot of people doing it DIY out there, probably without consideration for probably the health and safety effects they probably should, which is why it's often best left to the experts. But yeah, it's a simple formulation, obviously starting with ethanol. We've got some thickeners, neutralizing agents, and often some sensory ingredients, which is often at the discretion of the manufacturer. So if we start with ethanol, there's a range of different suppliers and sources, overseas and local. There's a heap here. There's two main manufacturers or producers of ethanol from wheat and sugarcane. And then there's a smaller industry, smaller distillers around using the byproducts of the wine industry, grape production. And I mean, this is sold into so many industries. For us, it's cleaning, beverage industry, food, fragrances in particular, and the fuel industry as well, so E10 fuel. And we have had huge issues, as have most of the other manufacturers, in procuring ethanol over the past few months. So starting sometime in March, the demand for hand sanitizers, probably late February, the demand for hand sanitizers started to increase, and we found it more and more difficult to get ethanol. Now there's quite a few different grades. You can obviously have pure ethanol, undenatured, and then you can have a variety of different denatured forms, which obviously contain ingredients that stop people using it for beverage or for consumption. So there is an excise tax on ethanol that's undenatured, and obviously we don't have to pay that if it's not for the beverage industry. So we do have that choice there, but because there was limited availability, we saw that there was a lot of people putting denatured ethanol onto the market in hand sanitizers. So you'd have things like methanol, isopropanol, industrial solvents like MIBK, MEK, and then bittering agents like denatonium benzoate. So there'd be a whole range of different denaturants that we could observe quite easily in other people's products and imported products in particular. So that was a bit of an issue, and obviously there were some reports of impure sources of ethanol where we're getting hints of grape must coming through in various competitor products, and overall the industry was a little confused. It was hard to find ethanol, and the source and the quality was all over the place. So there was a lot of work with the industry group that we have, lobbying government, trying to improve the supply and availability of ethanol in particular. And the strange and confusing story that we were getting at the time was that there was no issue with supply. And what a lot of manufacturers like us were saying was, well, why is it that we can't get it? And there was a lot of back and forth there. And in the end, supply did free up eventually, probably late into April, I would say, from memory. But in the meantime, for about 7 or 8 weeks there, we did have to find alternative sources, which was quite difficult and very competitive. And luckily, we had a number of relatively large distillers who supported us in that, and we were able to continue supply. Now, the next set of ingredients, thickeners and neutralizing agents. You can see there, they're often based on polyacrylate chemistry and alkyl substituted polyacrylates. So this was the other ingredient that was in high demand. You could not get it anywhere. Luckily we had quite a number of channels for different thickening agents that we could use. We saw a lot of other people were finding this really difficult to get. If you could get it, it was far more expensive, and we were going from an 8-week lead time to 20 weeks. And there were a lot of companies out there that just weren't taking orders. They just said, we are completely full. There's no possible way you can get any more. So over the period of about 8 months, we reformulated our products 7 or 8 times to allow for different thickening agents, whatever we could get at the time. This included various number of derivatives, sulfonated derivatives, various substitution mounts on the acrylate backbone, even some cellulosic gums. And that kind of flows onto some of the sensory effects as well. So we would find that based on the different thickening mechanisms, the viscosity and the rheology as well, so how shear thinning these thickeners were in the ethanol solution would dictate how fast it dried, how it felt on the hands, how the hands felt afterwards, whether you get a sticky or a tacky effect, and whether you would have a clear, transparent solution, or whether there'd be issues with clarity. Now, this was difficult for us, and especially in terms of marketing approach, because customers were confused. You know, people were getting a slightly different version of the product, and we did our absolute best to make sure that it was as close as possible to the original, but that wasn't always achievable. And we saw a huge variation in the quality of hand sanitizers available in the market in that sort of 2-month period. And prices obviously were spiking as well. So the last set of ingredients that usually go in a hand sanitizer formulation are sensory. So sometimes you might add a fragrance. We don't happen to. I mean, there can be some allergic reactions that occur with certain fragrances with certain people. It's often a small percentage of the population, but it does occur. But people do put fragrances in there. We've got menthol and citral down there. Got a skin conditioning agent, panthenol. Glycerin's a pretty common one that you can sort of get anywhere. People usually do that on home products, a bit of a humectant, just keeps the moisture of the skin. And then aloe vera has always been what has been popular during this period. And these are just some of the alkaloid components of aloe vera. So in that respect, you can get quite creative. And whilst we didn't see any real issues in supplying these sort of minor ingredients, we didn't really look at aloe vera too much, but we did hear that was in demand too. So from there, whilst we managed to get through all those challenges with formulating with the alternative thickeners and trying to source ethanol, which almost became a full-time job, the WHO formulation that was released, I think it was around the last coronavirus outbreak in 2009 or possibly earlier, the TGA created an exemption for ARTG registration for any product that used that WHO formulation. It was very limited to one ethanol or one isopropanol based formula, and it allowed anybody who was manufacturing that formula to sell it into healthcare, which previously would have required a TGA registered product, which was also extremely difficult to get because there are only limited number of suppliers, and they were obviously at capacity during that period. So whilst this was fantastically responsive of the TGA, it obviously did open up the market to almost anybody, really. Also, just on that, they originally specified pharmaceutical grade ingredients, and I think the feedback was probably that this was extremely difficult just due to a lack of supply. So they later amended that to be to allow food grade ingredients. So there were some practical challenges with this formulation as well. Mostly the fact that it was water thin. It didn't lend itself well to pumping. So the normal type of lotion pump you'd have for hand sanitizer products, you'd end up squirting it, it would splash, it would drips through your hand, drips on the floor. Sometimes the pumps wouldn't hold it if you had an automatic dispenser, all sorts of issues there. So we needed to supply different pumps, and we needed to change labels, reprinting. So there's a lot of other issues that went along with that. But I guess the key point was it was effective. You know, it's a way for the regulatory agencies to standardise what's out in the market, and at least exert some sort of control, I suppose. So packaging was the next issue that we faced, and again it was lack of supply. So what we found was that there is enough suppliers in Australia to make all of these, but the demand obviously had increased and they were unable to, and then they hit the repercussions of the China shutdown and probably Asia shutdown, where manufacturing was closed in those regions for a period of time, whether it be a few weeks up to 6 weeks. So the raw materials for these were no longer available. So they were seeing the delayed onset of lack of supply of raw materials like polyethylene, polypropylene, and various other plastics. For us, it's mostly those two, polyethylene and polypropylene. So often the bottles are made of HDPE, caps and pumps are often PP. And what we found was that we had to pivot quite quickly to almost whatever was available. So if it wasn't a pump, it might have been a flip cap or a push cap or a push pull or a spray, anything we could really get that wasn't just a cap, because we still had to make sure that it was safe for our customers to use. And again, this presented a lot of other challenges, which I'll let Amber continue talking about.
So this is an example here of one of our hand sanitizing products, and all the different packaging configurations we've had over the last 2 or 3 months. So on the left is our original packaging, and then we've had to have different options of that in our rectangle bottle with black or white pumps, sometimes a flip cap, and you can see on the far right, even a squeezy version. So we just had to do a few changes to the labeling, so just to ensure that they fit. For the example of the rectangle bottle, we had to make a front and back label, and this is when we would need to work with Damien and the regulatory and compliance team and just make sure that nothing's dropped off, so no hazards or claims that need to be on there gone missing. We need to have a bit of back and forth with the teams to make sure that each time we produce a new bottle, it's correct and it's following regulations.
And it does change the way we manufacture the product too, which line we can use, the method of labelling, and things like that.
Yeah, so there's different ways we've had to adapt to the changing market, and resizing the labels has been the main one. After that, we've had some issues with having to create a label without actually physically seeing the bottle, because we've been working from home or we've just had to purchase a bottle in order to supply customers. And the other thing would be the on-flow of that, so updating marketing material, making sure that the images of the products actually match what the customer is going to receive. And and then after that, producing marketing material to assist customers. So yeah, there's a lot of new competitors on the market, so educating them on the differences in our products, and yeah. And then creating signage to accompany the product. So there's an example there of you know, please sanitize your hands, something a lot of customers might not have previously had. They might have just had a product, but not also the prompt for users. And then recommendations for their COVID safe practices on where to use the products. So entrances and exits, transaction counters, voting rooms, meeting rooms, all of that sort of thing. Just a bit more education on when and where to use these types of products.
Look, it was something that would normally take maybe 4 to 8 weeks if we did it under normal circumstances. It's now being condensed into a few days or even a week, often without seeing the packaging beforehand. It would be purchased from overseas in large quantities with a long lead time, and you were having to, well, not guess, but it was a calculated risk. Printing labels at the same time as we were purchasing bottles. So like we've
specifically with the there was quite strict guidelines on what we could and couldn't say on the label. So that's where we had to get a lot of feedback from what we're allowed to say. Because with marketing, you do want to run those pandemic things.
So sorry, Amber, we are still struggling to hear you. I do apologise. It's really cutting in and out. We're catching some of it, but not all of it. So I don't know if the microphone's on one side of the PC or something. Why it's happening like this? But
I believe it's got an anti-amber frequency.
it must have. It must have. So yeah. Obviously, a lot of the information is on the slide, so I hope people haven't missed too much. But if there's any questions for you to answer, Amber, we might try and get you a bit nearer or put some stuff in the chat window and stuff. So I do apologise for that stuff. I don't know why, but it only likes Damien.
Yeah.
Okay, look, we'll leave it there. We're happy to answer any questions.
Yeah, look, and just on from that, a couple of questions came through while you were talking. So one of them was what is the purpose of a neutralizing agent in these products? Yeah,
okay. So often I probably should have mentioned that. With these type of thickeners, you do need to push it to the salt of the polymer to push the strands apart, I suppose, create that distance and create the gelling effect. So depending on what we neutralize it with, there's a range of different neutralizing agents we can use. But yeah, the idea is just to bring the pH up usually past the pK of whatever the acid is. And in this case, it can be anywhere up from pH 7, it starts to thicken up. I mean, if you're using the cellulosic gums, that's not the case. You do see a slight change in the viscosity over a broad pH range, but generally it's a different mechanism.
Another question on the thickeners was what type of thickeners are suitable for the ethanol IPA formulations?
Yeah, traditionally the carbomer and polyacrylate style thickeners are traditionally used. I think you get a good combination of affordability with hand feel, ease of use, clarity as well. First, you get transparent solution. There are a range of other thickeners you can use, and we've had to look into that. We started looking into that when this happened, obviously because availability of raw materials was scarce. So there are a number of other options that we went with that we wouldn't normally have considered. But yeah, normally it's those carboners.
Did you want to add something to that as well, Greg, on that point?
No, I think Damien's answered that.
Okay, fair enough.
Very well, but the question that was allied to it is how much latitude does the TGA give us in terms of what thickeners we're using? And the answer is yes and no. What they have done in this current COVID period is they've put more resource into expediting change applications. So for us, where we've run out of one particular carbomer, we have access to another one. So what we did was we actually then had to redo the formula and pull the thickener out. So it's now water soluble. It's very runny. That's a bit of a dispensing issue. But the TGA allowed us to fast track that reapproval with a minimum of data set. So that was actually really good responsiveness by the regulator. But if you were to bring in something totally new, that would slow everything down unless it was a material that was already on a pharmacopoeia somewhere. So if it was on the British pharmacopoeia or the US pharmacopoeia, one of the European pharmacopoeias, yeah, they'll have a look at it. But if it's a material unknown to Australia, that's going to slow you right down. And of course, if you're not inside the TGA bandwidth and you want to do that, and you want to bring in a thickener and it's not on NICNAS, well, you're in a world of pain. There's no shortcut for that. It's just a world of pain. So we don't have time to do NICNAS just at the moment.
So, you know, when I go to the local Kmart or something, the sanitizer is quite thin. I wasn't really expecting that. Are there more prevalence of those on the market due to these reasons then? Is that you wouldn't see so many of those normally? All be the pump action thickened stuff?
Everyone's making money.
Yeah, fair enough.
Some of them are going to lose money, let me say.
Yeah.
The guys who relabeled their gin incorrectly the other day, they might have a bit of a... Sorry, Damien, it's your presentation.
No, we're all good. No, you're okay. We're really open to the open floor now, so I'm just going to have a quick look. So, were refill bottles an option at all in terms of the manufacturing side?
Depends how desperate we were, I suppose. We did manage to just keep up. So maybe we didn't get to that point. I mean, that's a logistical issue, logistical nightmare. You know, collecting them back, you know, how do you ensure that they're washed out correctly? Do you have a license to wash them out and get rid of the waste? And there's a whole heap of other considerations with reuse of bottles that isn't just about bringing it back, filling it up, sending it back out again. You say the same, Greg?
Oh, I agree. Reusing bottles microbially is just a nightmare. But if you want to create superbugs, that can be a really good way of doing it, because you know, you've got sublethality as an issue all the time. And, you know, as I said in a little talk, I mean, the bugs want to survive. That's what their intention is, you know. So, and logistically, how do you then handle all the wash solutions and the water? I mean, oh my God, the waste is just out of proportion.
Yeah.
Yeah, look, we've had talks in the past in some of our seminars where we've talked about manufacturing medicines where you can have a liner that you throw away because then you don't have to do all your cleaning validations. You know, actually just might be better for the environment. But anyway, different talk. Someone's asked about why hydrogen peroxide is needed at all in these formulations.
Oh, I'll answer that. That's hilarious. So that formula is, if you want to destroy your skin, the WHO formula.
Oh, okay, fair enough.
And the reason they've got the hydrogen peroxide there is it's a formula designed for the third world where they've got no expectation of water controls. So the first thing you do is you fill whatever part of your tank up with water, and then you're basically disinfecting your water with an excess of hydrogen peroxide.
Right.
And they use the hydrogen peroxide because they can't use chlorine in these solutions because it will completely destabilize everything. So you end up with an excess of hydrogen peroxide on the skin. It's a good antiseptic, but
it really is hilarious. I read the WHO documentation on how they developed this, and they said it's mainly there to kill, if from memory, fungal spores that might be in the water for 2 or 3 days.
Yeah. Oh, and single cell amoeba. So you know you're worried about the parasite.
It takes it into question. If you put pharmaceutical grade ethanol and then just put pond water in, it's not really the right thing, is it? You know. But anyway.
Well, the TGA would have something to say about that, I'm fairly.
Pally, there's a question for you. Someone said, I'm confused as to what is a soap. Can I use anything? Soap?
Well,
there's some soap-free things been on the market, and then people are a bit confused by the labeling, I think.
Yeah, so I was inundated by emails like this after the Twitter and the media storm started. Like, so what? Yeah, I can go to Coles and buy the soap-free, soap-free soap. So basically, okay, so the two main types of soap you can buy are the synthetic soap, which is normally SDS or SLS, or sodium lauryl sulfate, is the main ingredient. There's usually lots of other stuff in there. Or you can buy natural soap, which is of course chemicals. And what you do there is to mix fatty acids and sodium hydroxide essentially. So that's how our grandmothers made soap or whatnot. Now, because there's this sort of chemophobia out there, some people worked out that you can market SDS soap-free products, and that's really the soap-free stuff is the SDS-free stuff. There's no sodium lauryl sulfate in it or SDS in it, right? What they put in is sodium betaine, laurate, whatever. They're very, very similar surfactants, you know, like there's an ethyl chain hanging off there or whatever. It's pretty much the same stuff, and still soap. For me as a chemist, still soap is just, you can market it as SLS-free and call it soap-free soap. But I guess you've got to read the ingredients. So yeah, I was inundated with emails. People say, this is what the label says. And well, it's still a surfactant of a kind. But Greg, I'm with you though, that we don't really know which of these surfactants are better or worse than others. And of course, for me as a chemist, when I look at a quart, they're really just surfactants, but they're much better at breaking up these membranes. But there are some health concerns with them, right? I guess that's the downside of the quart. So,
well, I think with all the disinfectant molecules, I mean, they all react with biological material, which means they'll all react with us. I want to go back to one thing you said, and just to emphasize as well, Paulie, we didn't have time to cover all the technique, but we absolutely agree that the first thing you do before any disinfecting process is you've got to do cleaning. If you don't remove the soils that are there, your disinfectant has got a massively elevated risk of failure. With biofilms, work we've done and published has shown that chlorine, for existence, when you put Staph aureus into a biofilm, Staph aureus will normally be killed at 1 milligram per litre level. You put it into a biofilm, it will resist up to 20,000 parts per million.
Yeah.
And, you know, we talk in terms of antibiotics. Once these bacteria are in a biofilm, they're usually 200 plus times more resistant to the antibiotics. Part of that is just physical access of the chemistry to the molecule, the chemistry, actual drug. And so that's indeed part of the whole process. So the use of a soap is absolutely right. And in fact, my favorite term is neutral detergent. It gets bandied around. And it's like, are we talking charge or are we talking pH? There's 9 variables there. Which one are we going for? And it's in the literature. This is the terrifying thing, the medical literature. It's everywhere in the nursing literature, and it's an undefined nonsense.
Absolutely. Yeah. But what quartz are though, I mean, they act non-covalently because they just positively charged surfactants. So they're unlike chlorine, which chemically reacts with your fatty acids or your protein. So there is a bit of a difference there. But the only thing I should say, I've read now, is that some people think quartz can still have some long-term effects on health, and that is sort of why there are countries which are trying to ban use of quartz in skin products. But I don't know whether that's a bit of an overreaction or not.
I think it probably is. If you look at the amount of response to, say, the amphoterics like chlorhexidine as antiseptics, I mean, they've actually got a worse profile, and yet they are the standard point of use for antibacterial hand soaps globally. And, you know, standard antibacterial hand soap surgically used is usually the 4% chlorhexidine, 0.5% of an iodophore, or an 80% plus, usually 90% alcohol solution, ethanol or IPA. So once again, you're getting into an incredibly vigorous sort of area. If you look at in terms of the potency, in terms of molecular potency, chlorhex is going to be used at 2% as a classic sort of response. The same response you'll get from a quad at about anywhere, depending on the formulation, anywhere from 0.15 up to about 0.3. So, you know, you're an order of magnitude lower in concentration. And then you also get into the difference between the single chain quats and the dual chain quats. And look,
am I right that, say, you know, with the virus on one hand, you can quite easily destroy it with soap or something like that, whereas you can't, most bacteria you won't easily kill with soap, which is where you need proper disinfectant. But the flip side of that is that when I look at the molecular structure of chlorhexidine and viruses, it's really good evidence that chlorhexidine itself destroys viruses? Isn't it really just the other stuff which is in the formulation that would destroy a virus? Because the molecular action of chlorhexidine is very well understood with bacteria, but I can't see how it should interact with the virus.
Yeah, well, there's no data out there because chlorhex is almost exclusively used as an antiseptic. Therefore, the claims are illegal. You know
exactly. Yeah.
And for those that follow the web, Purell Corporation in the US is currently under notice from the US FDA for illegally marketing their products with antiviral claims, and so they're in a current, put up or shut up time. So. That might get a bit exciting for Purell Corporation, which is a privately held business in the US. So look, the whole area about this formulating science, see, one of my father's best patents, he was a life member of RACI. It was actually not so much about the disinfectant molecule, it was all about the surfactant package around it and getting surface tension into the right area. It was a brilliant patent, the products had great life. We still sell the product nearly 35 years later. And the disinfectant molecule is not so popular, but the principles in the surfactant package, which are all about wetting and solubilization and approach to the surface of the bacteria or virus, is all about fundamental surface chemistry. You know,
cool.
Part
there was a question around the kind of antiviral and antibacterial wipes, and really that they were labeled with Lysol and benzalkonium chloride. Are they valid to say that they can do anything with coronavirus, or do you know anything about those ones?
Well, I know that Reckitt Benckiser, who own the Lysol brand name, I think it's Reckitt's, they've already got their product on the Australian list. The easy thing to do is just for when people get the browser, if you want to have a look, just hop onto either the US EPA website or the Australian TGA website, and all the brand names turn up.
Yeah.
What was the active ingredient there?
Well, Lysol usually uses orthophenylphenol, but sometimes some versions of the product are using triclosan. So it's phenols of various sorts. Traditionally, of course, if you go back far enough, you end up with PTX, parachlorozole, which is the additional Dettol solution. So they're organic phenols of various sorts.
And someone's asked about, ethanol versus IPA. It's, obviously we talked about swapping out ethanol or some of the concerns. How do the other alcohols work in comparison?
They all dehydrate the cell. They basically work on isotonic pressures. So,
okay.
And they are different because there's a slight difference. I can't explain the difference, but generally they're a couple of % apart to get the same level of efficacy. But basically one's coming from wheat and barley and the other one's coming from corn. The Americans grow a lot of corn, so they use a lot of IPA, and Australia's got lots of wheat and barley, so we tend to use that. And sugar cane, of course.
There was another explanation in the WHO documentation too, which is that in certain countries, ethanol is culturally or for other reasons banned. So you would use isopropanol.
Yeah. Right. Yeah, it's certainly hard with the sanitizer in some of those Arab nations would be very difficult.
Well, also in Scandinavia, where I come from, ethanol tends to be taxed to death by the government, so you wouldn't use that if you can avoid it.
Yeah.
Maybe Pally, same
here. Damien, there was a question around raw materials. Obviously, there would have been an expectation that you'd struggle to get hold of stuff. How much can be sourced locally, recycled material? I think you talked about distilled material and things like that. How much can we get locally, or how much did you manage to find locally?
Do you mean ethanol, or are they talking
anything really, so you can make your product?
I guess it depends how well connected you are into the supply chains. We've been around a while, so we've got some pretty broad connections, as I'm sure Greg does too. So we had a lot of avenues to check and people to talk to. I think we would have sourced 100,000 litres at least from alternative sources that aren't from our existing supplier.
Mm-hmm.
And then obviously we diverted back to one standard supplier after it restarted, and they were able to continue. So, but yeah, we were extremely grateful for that, and actually quite surprised that it was available and that we could get it.
Yeah, I think obviously once the pandemic hit, everybody suddenly realized, hoping they could get stuff locally. But when you've been years, even outside of just disinfectants and all that in the industry of getting everything from abroad, some things you just don't have the option, do you? Really, you just don't make it anymore, or it's too expensive, or all those things that have occurred over years. So yeah, it's an interesting one. Someone mentioned about hydrogen peroxide being expensive. Is that right? As an ingredient compared to the others, is it an expensive ingredient? Just
not there in great concentration, so it doesn't.
Yeah,
really...
okay, doesn't matter too much. Cool. Someone's asked about how effective essential oils would be without the alcohol.
Do you want to take that?
Well,
is that not at all, or I'm just going to guess the answer.
Essential oils are fantastic, but you've got to be careful about what you wish for. If you want to find the most toxic thing in the kitchen cupboard, it's usually eucalyptus oil. Don't drink it. Look, tea tree oil, we did some work on tea tree oil many years ago. There's 27 different fractionations, 2 or 3 of which are really potent biocidally, but they evaporate so quickly that if you don't store it in glass or metal, it's basically useless. So when you're formulating in high density polyethylene or even a fluorinated bottle, you just lose all of the active materials. Funnily enough, tea tree oil is one of the products in the US, this is a bit of trivia, that's actually grandfathered onto the Federal Register in the US as an antiseptic without ever having had to have produced data, because it was exported from Australia before the promulgation of the Federal Food and Drug Administration. So you can buy tea tree oil in the US as an antiseptic, and it's never been through the monograph. It doesn't need to go through the monograph. And it is a pretty good antiseptic within constraints. Again, don't drink it. Apart from the taste, it'll kill you quicker than Panadol. So
yeah, fair enough. Damien, are your products sold universally or just to business and health services? You may have covered that, Amber. I apologise if that was covered already.
That's okay. Look, we do primarily, we do sell B2B. We're quite broad actually in our scope. We have a few different companies within the business and brands that we sell into. So we do have a business called Cafetto that sells exclusively into the coffee industry, for example. That's international, and the variety of products we sell there is completely different to what we're selling in the dominant business up here. So we sell directly to the public through our home products website as well. So we've got avenues everywhere, really.
Okay, cool. Sounds good.
I was going to pick up on one question that's on the chat thing, if I can, Scott. Asked about one of the 27 products on the TGA list is described as blah, 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 blah. Well, it's one of the anomalies of the law. Is the TGA are really vigorous at pursuing the evidence insofar as performance, but the rest of the claims burden actually falls outside of their legislation. There's some comedy in there about 100% effective and safe. And the funny thing is, the word they can't use is the word safe. They can't say 100% safe. That's covered in the legislation. But if they want to say chemical free and grows on trees, the TGA tend to have a less rigorous view. That tends to turn up, however, in the ACCC area. So the ACCC then take a very dim view of claims that are beyond what might be regarded as marketing fluff. And so there's a bit of artifact in there.
I think there's too many chemists probably listening that don't want to hear the phrase chemical free, you know what I mean? But that's all good. Look,
I'm sure Damien and Harley are the same. So.
Well, look, we're at our kind of time limit. I might just ask, was there any question you guys wanted to ask each other, anything you wanted to add? Otherwise, I was just going to possibly ask the first question that was asked, but around imported goods. Otherwise, is there anything you wanted to cover off before we sign off, you guys?
I'll just answer one last question about what happens after the pandemic. Depends on whether we get a vaccine. So, you know, this virus might be here for a little while yet. And look, I think the principles of good hygiene apply no matter whether you're in a pandemic or not. And in hospitals, the issue will still fall back to hospital or healthcare associated infections. I mean, the infection rate in Australia, which is still not published, possibly runs as high as 1 in 10 surgical patients in public hospitals around Australia. That's absolutely unacceptable. The US started publishing when the rate was around 6%. They've dropped it down. But all those principles of hygiene, wash your hands regularly, clean and disinfect surfaces that are touched regularly, those issues don't go away.
I did a bit of a question for you, Greg, or thought, because you talked about the testing requirement and how strict it is in terms of what you're allowed to say. And in my screening of the scientific literature also, I find it very wanting. I mean, what is done there, I think, is pretty ordinary. Can I put it that way? Is it possible that when we come out of this pandemic that we can seriously revisit all those issues and put some effort into better, more rigorous, both and relevant in vitro and in vivo tests to work this out? Because I think like we just lost the boat there the last 50 years. I think as a society.
I agree, Paulie. Two things. First thing, the tests that are used in the standardised criteria are all fully validated. There's a long history. But as I said to you privately, and I say publicly, that won't turn up in the peer review literature. So that length and breadth of history is outside of most people's sight. And, you know, companies like us who have been doing it for 30 years, you know, we just know what to expect. The other thing about microbiology is and the second thing is, it is live science. I mean, you know, if you run an experiment in a chemistry lab, you would expect that if you run it correctly, it will turn out each time and every time to be generally within boundaries, give you the right outcome. With microbiology, there's always a bit of give and take because the bugs are intending to survive. And so even using standardized bacteria in labs, you regularly have to test that the standardized bacteria in the lab has not evolved. So it evolves to a more disinfectant resistant status.
I guess it's worse again with viruses, right?
Absolutely. And look, in terms of, we don't have time to get into it here. I'm happy to talk to you privately about virucidal testing, but what you had up wasn't quite on what happens with the standardized tests, because depending what the virus is, depends on your recovery system. So some of them are using cell lines, something like some of the others like influenza, you use live eggs. I mean, you inoculate the virus into eggs. Some of them you're exposing it where the virus is dried as a dried droplet, if you will, on the end of a stainless steel.
Yeah, that's the one I read most on.
Which is then inverted, you know, immersed in the disinfectant. But there's an array of things that happen depending on what the virus is, what the application is. It's only when you come to do the regulatory testing that you fall back to these standardized tests. So what you see in the literature is just awash with stuff that may or may not be valid. But in the regulatory framework, you're really into the hard end of validation and
actually I see there on the chat line for Patrick, there's a related question. How do you detect the viruses and surfaces? And if I may, my neighbor here is Justin Gooding, who works on all sorts of sensors. And we had a forum about that a few months ago. And you see, the problem there is that if you detect just for the RNA, you don't know if you're testing for an active or inactive virus.
Absolutely.
And on the other hand, if you try to use antibody-based detection, you always get the problem with false positive. So it's actually a really hard problem.
It's a very difficult science. And the other thing is, of course, viruses are not very big. And if you're doing something that's even a high touch surface, the surface area ratio to the virus means straight away you get into sampling error.
Exactly.
So, you know, type 1, type 2 sampling error everywhere. And, you know, when it comes to something like cleaning science, that's why in one sense they're all wandering around with wands waving them everywhere, because they're hoping they get to every surface. But it's just a nonsense. It's a nonsense. You could run an argument that it's a great way of proving the null hypothesis. But anyway.
Now, look, I think that's good. Look, we have slightly run over time. Thank you all. I think that's a good place to end there. we do have one of our microbiologists on the line. I won't mention her name, but she's basically said microbiologist rule. So obviously we get that micro in, and it's way too hard to, way too hard for me to understand as a dodgy chemist. But no, thank you all for your time. I apologise, Amber, of what happened with the sound. That's Zoom for you, I suppose. I apologise that it was difficult to get your points across. I hope too much wasn't missed there. But thank you all again. Thank you, Greg. Thank you, Pally. Thank you, Amber. Thank you, Damien. Thanks everybody for listening.